So the question revolves around a class of medicines known as thiazolidine-diones, or TZDs. There's more than 10 million Americans on it. For a while, I would say it would have been the mainline therapy for type 2 diabetes, or one of them, if you could afford them. There have been some issues of side effects. One of the side effects that's unpleasant for the patients is they get fatter. And as an example, you see many patients, I'm sure Gordon sees the same, that if they're on TZDs and insulin, they'll gain 50 pounds of fat. So we're not talking about a small amount. And these drugs have a really profound effect that they cause redistribution of fat cells. And our notion, and we have some data, is that indeed they work by lineage formation, and we hope to put together a manuscript in the next couple weeks even to submit in that regard.
Thank you. Yeah.
And of course, that highlights the notion that being in the blood vessel wall makes them potentially very therapeutically accessible and maybe an easy place to target with small molecules.
So I had a question that's kind of directed at all of the speakers. We heard a lot about the generation of patient-specific iPS cells for studying diabetes and strategies for reintroducing cells that might produce insulin into diabetic patients. And we also heard about the possibility of intrinsic differences in the differentiation potential of these cells and about effects of the environment, for instance, adipose tissue, on the functioning of the cells. And so I guess I was interested in your thoughts as to considerations as to how one chooses the donors for the first experiments to test out these ideas, and who would be the appropriate recipients, or what would be the considerations for choosing the first recipients of transplants, taking all of these issues into consideration.
I'm sure you all have opinions. None of you are shy. All right, Ed, go.
Yeah.
Can I call on you to lead off?
I mean, I think where we would go would be for sort of a stable type 1 diabetic population to start with. We probably would look at their HLA type and see how it matches or doesn't match with the HLA type of the stem cell population that we're using as a source, just to give us an added safety advantage with regard to if the, say, device ruptured, that the immune system would be active in rejecting it. But I would say that, it would be, most likely a population that was stable diabetics, so that in case there was an allorecognition response to the cells that were transplanted, that they might not need an organ transplantation in the later stages of the disease because they didn't have complications.
Can I get on to another safety issue, and that's the mesenchymal stromal cells? Seems to me there's a certain amount of anxiety about that, and maybe, Armand, you could lead off on that. I'll bet some of the others have opinions too.
Yeah, this is an issue that a number of us are concerned about right now. There's a big difference between the ability of murine MSCs to undergo transformation and human MSCs. The mechanism appears to be related to the ability of those cells to undergo senescence and then to progress and transform. My own experience is that it's very difficult to transform human MSCs or to have them transform spontaneously. There have been some reports of this in Europe, and it's certainly a matter of concern. One of the issues may be related to the number of doublings that these cells undergo. But most of the studies in which the doublings have reached about 20 or so appear to be reasonably safe, or at least safe in terms of karyotype stability, non-tumorigenicity, and so on. It is an issue, though, and I think we need to watch it carefully. I think the release criteria should include karyotype analysis.
Directed to Baetge and anybody else who wants to answer. The device you're using has reasonable hope of protecting against allogeneic immune response. What are your thoughts about protection against the autoimmune response in type 1, and would iPS cells give any benefit for type 1 diabetic?
Yes, this type of device has been used in the past in the non-diabetic model with not pure islet preparations, but with transformed islet cell preparations, and has been able to show in a short-term study that this type of a device could keep those cells alive and correct the diabetic state. So there is some limited data that suggests that this sort of device could protect against allo as well as auto. Now, with regard to IPS, you certainly, will, if you use an IPS cell in the device, an IPS-generated islet, you're going to have less of the allo situation, but you're still going to have potentially the auto situation. So again, you're going to have to protect against auto in a type 1 situation. Now, in a type 2 situation, you may have a more favorable environment where you don't have the auto and you just have to protect against allo. So theoretically, in a type 2 situation, an IPS-generated islet, if safe, could potentially be delivered in an unencapsulated way and still be effective. Now, still a whole lot of work has to be done to say how identically these IPS cells will be reflected by the immune system of those people that you put it back into. And right now, I don't necessarily, there is no answer to that, but that really has to be resolved before you can really say that.
Well, if hands don't shoot up, I got more questions. I want to get you some thoughts about how fast beta cells mature, because, in normal pregnancy, don't they appear at sort of 10 weeks and somewhere around there? And so they're sitting there for another 30 weeks. So you take your ES cells and you put them in a mouse, and you get maturation over some period of time. And you treat the mice with streptozotocin, so you may knock off a population of older cells selectively. And there are experiments where people have taken human fetal pancreas of different ages and transplanted them into mice. And I can't remember exactly how long it takes those to mature, but my impression is that when you take something that might be at 15 weeks or whatever, and you put them in a mouse, that they actually mature much faster than, say, another 40 weeks. So it's sort of a complicated question, but I guess the sort of answer is if you take a beta cell that's formed at 17 weeks and you put it in a normal environment as opposed to the in utero environment, which is very different and has a lower blood sugar, for example, among other things, would you accelerate the development of that beta cell? And I know Doug and Ed have thought a lot about that, so I'd love to get some thoughts.
Well, I'll just start out. There's a lot of things I can say about what you just asked, but in the studies that have been done, say, by Raphael Scharfman, where he's taken human fetal pancreas and placed it under the kidney capsule, I believe that those studies take between 4 and 6 months before you have real good islet function out of those. And the graft grows quite considerably. Now, in that situation, you're transplanting an onlogger, complete pancreatic onlogger dissection from the fetus. So you've got a lot of the mesenchymal elements as well that are coming along, and that then pancreatic epithelia or small number of islet cells that are there at, say, 6 to 8 week post-development, post-conception, are coming along. So that's somewhat different from when you do it in an embryonic embryonic stem cell-based approach, where you're making primarily an enriched population of the epithelium without the human mesenchyme that's normally present there. So this could either speed up the process or slow down the process. And for us, if you add up all of the days, it's about 17 days in vitro or post-conception, and then it takes minimally 30 more days to see the first C-peptide in the blood and the first small pockets of islet cells. And by about 90 days do you get good islet function. So it actually, when you add it up to when it occurs in normal human pancreatic development, is pretty similar. It's about 100 days or so.
Ed, what were your thoughts? Or Doug, I mean, what do you think?
Maybe I'll just add a bit to that. I don't know the literature as well as I should, but I haven't seen any paper that I think securely establishes some of the things you said about this maturation process. So I would frame it slightly differently. I don't know of any developmental decision that takes 100 days, particularly if we're talking about one cell and not the progeny of that cell. So I would have thought the challenge would be to find a molecular correlate of this switch from being non-glucose responsive to glucose responsive, and find out really what that is. It could be something quite trivial, like the phosphorylation of a protein in downstream of the GLUT2 transporter. So I guess just to repeat myself, I think the simple minded experiment would be to take cells that are and are not glucose responsive and try to find out can you identify a molecular correlate of that difference? Because I just cannot believe there's a developmental decision that takes 100 days.
Yeah, that's what I'm sort of, whether there's just some terrible, clock that will just doom us to have to wait. I mean, when you put immature neonatal pig cells into mice, it really takes weeks. It may take 2 months, 3 months for them to mature. So that's sort of similar to Sharpman's experiments with the fetal pancreas. Well, in any event, somebody should figure out some rigorous way to address that.
Jacob, I have a question for you. There's a number of congenic strains of mice with the NOD where they don't become diabetic. Have you tried to look at some of those to see if those animals that don't or resistant to diabetes are easier to be able to get their cells to work?
That's a great question. As we're trying to figure out if there's correlation between some of the loci associated with diabetes, whether that would have anything. We haven't done that as extensively, and we're still, Mike Wilson is doing some of this analysis. But from the couple of strains we've tested, the congenic mice, we cannot really tell that there's a correlation. It's probably something else, but at the moment we're not sure.
My question is for Dr. Graff. I was struck by the pictures of the mural cells around the vasculature and then the lack of mural cells in some pretty vascular tissues like retina and showed kidney and cardiovascular. I was wondering if you looked at the liver because, certain drugs cause massive toxicities of steatosis in the liver. And I was just wondering, what your thoughts are if you've thought about do the cells migrate from vasculature that doesn't have mural cells to now the cells moving to the liver, or are compounds perhaps inducing mural cells, transdifferentiating cells in the vasculature of the liver, and perhaps if some of your organisms could actually be models of steatosis.
First, I apologize if I suggest the notion there aren't mural cells. There were mural cells. They just don't express this marker. So those other tissues we chose specifically because they're rich in mural cells, or they have diseases of mural cells. For example, in the retina, those mural cells are thought to be an important target of eye disease that forms with diabetes. So we chose those specifically because they're chock-a-block full of mural, but yet don't express the stem cell marker. So they, in fact, do. The liver also has these stellate cells, probably as you know, that are mural-like in composition. A lot of the deposition of fat in the liver in fatty disease is liver parenchymal cells and is probably an SREBP-dependent process rather than the formation that you see in adipocytes.
So one of the things really from our model that struck us perhaps, could this be happening in the human? So kind of obviously immediately went for it, and it didn't really work. You can see that this in 129s and NOD and basically all mouse strains and rats that we've tested, there's a similar behavior. And so instead of going up in evolution, we actually now have some interesting stuff in zebrafish, for example, embryonic stem cells, but that's aside. But regarding differences between, if you want to take human and mouse, though APSCs and human ES cells share a lot of features, there are still differences. For example, human ES cells, many of the lines do have the X chromosome reactivated, like REX1 locus is expressed versus APSCs shut it down, FGF5 expression. So in a way, you know, a human ES cells still resemble mouse a lot. So it still could be some species differences and difference in development after all. But I think it will still help us once we know the genetic elements involved in this. We'll feel more comfortable to know what we are dealing with, basically.
There's another closely related tissue that's often discarded, that's the gallbladder, and it has a lot of similar features to ductal epithelium and pancreas. And I wondered what your thoughts were about using that tissue for potential reprogramming.
Well, there are claims for a number of stem cell-like cells in the common bile duct and in the intestine that could be reconverted or converted into a beta cell. So I think those are worth looking at. I think my comment about clinical intervention was more like if you aim for the perfect solution, you'd want to put the cells in the pancreas. And so let's step back from that to say that right now we're comparing 10 blood glucose checks and insulin injections to the perfect solution. So I was quite intrigued by Ed's proposal that, well, let's just start by putting it subcutaneously. It may not be as good as having the cells in the pancreas, but it could still be 10 times better than glucose monitoring and insulin injection. So I think we should be open to a whole range of things, always keeping in mind we want to aim for perfect euglycemia, but it should be a relatively low hurdle to get better than glucose monitoring and insulin injections. Ed, I don't know if you want to add to that.
Yeah, I would agree with what you said. I mean, we obviously want to go into a site that we think the best site is to be in the portal circulation, so the insulin goes to the liver first instead. But most or the majority of all diabetics are treated today by subcutaneous insulin injection when they require insulin. So it does work. It may not be the ideal physiological way in which to give the insulin, but at least it's a start, and then we can move from there.
So taking a step back and considering the point you raised about the relation of the gallbladder, let's say, and perhaps its developmental history in relation to the pancreas. I mean, I think one of the ideas behind exploring hepatocytes or gallbladder cells is that they come from a very similar domain of endoderm progenitor cells, and so it might be easier to reprogram cells from those domains into pancreatic cells and beta cells than going from somewhere else. But, and there's some new studies, I think from Jim Wells' lab and others, that the gallbladder and ductal epithelium, the bile duct epithelium might be closer to the pancreas in specification in early development than parts of the liver. On the other hand, the liver is big. There's lots of hepatocytes. And so it's thought that that might make it easier to convert some of the cells. The liver's well connected to the bloodstream, and there's some good mouse paradigms now for interconverting some of the hepatic cells to, let's just say, insulin-positive cells, not necessarily great beta cells.
Yeah, I think that this has been very good, and so thank you all very much. And again, thank you to all the speakers and to the audience.